Good evening, everyone. Good evening. Good evening, Momita, Amrita, Vihan.
Good evening, ma'am.
Vihan and Amrita, good evening.
Good evening, ma'am.
Yes, Vihan, how are you? Vihan? What happened to your microphone? Okay. It would be nice, at least 3 or 4 students only here. So it would be nice if you all turn on your microphone and give me the response, ask questions. So all that. Okay, let's have the recap of the previous class with respect to RNA. So when we say RNA, what are the different types of RNA, Amrita? Amrita, what are the different types of RNA you learned? You understood? Amrita, are you there? Yes, Amrita, that's okay. What all the different types of RNA? We did RNA, right? We understood RNA also. What are the different types of RNA? Yes, anybody? Any one of you? What are the different RNAs, you know?
mRNA, tRNA, and rRNA.
rRNA. So which one is maximum abundantly present?
rRNA.
rRNA is abundantly present. Where is it present?
In the ribosomes and nucleolus.
Ribosomes and nucleolus. Okay, so rRNA are of 2 types. Which are they? How many types of rRNA you know? Okay, how many types are there? Okay, which rRNA is present in eukaryotes and which one is in prokaryotes? Based on the sedimentation coefficient, which are the different types of RNA? Basically, there are 4, 28, 18, 5.8 and 5. So these are the 4 types of eukaryotic RNA and okay, why are we talking about rRNA or mRNA and all this? So how mRNA is produced, formed quickly. So if you have the complete information about these things, then it is easy to go ahead with our next concept. Okay, okay, all about rRNA, Momita answer. Okay, Momita, tell me what are the functions, main functions of these rRNA?
Ma'am, rRNA, it helps in the process of translation. It's present in the ribosomes.
Exactly, binding the protein molecules. Okay, so they help in translations, binding the protein molecule. So they provide the binding site for RNA, tRNA. Okay, so what else are RNA's function? That's all. Okay, So coming to tRNA, what are the main characteristic features of tRNA? This is a very, very commonly asked question in CBSE. Over to Varshini, Amrita Varshini. All about tRNA. Can you please go on answering? tRNA is transfer RNA. And what is the name given to tRNA? What name have you given to tRNA? Amrita Varshini. We discussed all about tRNA, isn't it? What is tRNA? It's called as
soluble
yeah.
RNA that has a role as an adapter.
Adapter. What is the, why do you call it an adapter? Adapter, what it does? What is an adapter actually? Why do you call it an adapter?
Ma'am, it binds to mRNA, so it's easier for the amino acid to join the polypeptide chain.
So it has a sedimentation coefficient 4s and small molecules with around 80 nucleotides or 90 nucleotides, that's all. So is that single stranded or double stranded or multiple stranded? What is that? How many strands are there in tRNA? Go on answering. Is it single stranded, double stranded, or what is that? tRNA is also single stranded. Please remember, this is also a single stranded. Then why, what is the actual shape of tRNA? Why it appear to be folded in a clover leaf structure? Why does it appear to be folded like a clover leaf structure? What is the reason? Amrita Varshini. Why? Anybody? This is all CBSE questions, hot questions I'm asking you. What is tRNA? What is the name given to that? Why does it appear to be folded in a clover leaf structure? Why? Some of the nucleotides of this, that is nitrogenous basis of these nucleotide, get modified. So they get modified into pseudouridine, dihydrouridine, or ribothiamidine. So you can see all these loops there in the tRNA in your picture. So pseudouridine, dihydrouridine, inosine, so all these. So because of this, they appear to form the coiling. Okay, these loops. So this cause coiling of the single-stranded RNA, tRNA, into clover leaf or L-shaped structure. So in the three-dimensional view, so it appear to be L-shaped structure. So which is a clover leaf model as given by, who gave this model? Anybody? NEET question. Who gave this model? Cloverleaf model. Who gave this model? Cloverleaf model.
Holly.
Hawley. Okay, so Hawley gave that cloverleaf model. So what do you find in the 3' end of this tRNA? Quickly, whatever, go on giving me the answer. What do you find the 3'? What do you find at the 3' end? All these important to proceed now. Proceed further. Yeah.
Amino acid attachment site.
Amino acid attachment site. So opposite to that end, look into the picture. It's given in your NCERT, right? Is it given or not? Yeah, amino acid attachment site. Opposite to that site, what is there? Opposite to amino acid attachment site. So at 3' end, what is at the opposite site? Opposite loop is for anticodon loop. So what is that anticodon loop? It has 7 bases, out of which 3 bases form anticodon. So NODOC, NODOC, anticodon. So what is that anticodon? So let's understand. So, and there are other 2 loops, variable loops are there, then pseudo-uridine is there, then dihydroxyuridine is there. All these are different modifications of the nucleotide. So which forms the loop and coiling of this tRNA into this particular structure. So this much is the information about tRNA. So what is the other RNA? Now, what is the function of tRNA basically? It's an adapter structure, adapter molecule. What do you mean by that? So it helps to attach and transfer the amino acid to the ribosomes for the synthesis of a polypeptide chain. Okay, so different tRNAs will be there for different amino acids. Is that clear? Okay, sometimes some amino acid also can be picked up by 2 to 6 these type of tRNAs. So what do we understand here? tRNA plays a specific amino role here, important role. Okay, so for the transfer of these molecules to polypeptide chain. So for the growth of the polypeptide chain. So we are talking here about codons and anticodons. What are they? Codons and anticodons. So codons and anticodons are found in DNA and messenger RNA. Okay. So anticodons occur in tRNA, codons occur in DNA and messenger RNA. What is this codon? So let us understand that genetic code now. First, before moving ahead with the next concept, we need to understand genetic code. So what is this code all about? We are talking here about the formation of protein. When we say protein, protein is a, what is protein in biomolecules? According to your biomolecules, what do you mean by protein? What is a protein?
A polypeptide.
Protein is a polypeptide chain. So how is this formed? What are the monomers? Protein is a polymer of amino acid. Can we say that? Can we say that? Yes or no? Can we say that protein is a polymer of amino acid? Yeah. So that means what are these amino acids? What are these amino acids? What do you understand by that word? So let us understand now with respect to amino acids, and then I will move ahead with, before going to genetic code, let's go with messenger RNA, which is not understood. What's a messenger RNA? Quickly. What is messenger
Can you
RNA?
share the screen?
I have not yet started. I'm doing the recap of the previous class, so I'm going to share now. So I'm just asking you the previous knowledge. So we are done with messenger RNA also. What is a messenger RNA? Quickly, any one of you? Vihan? Vihan is very smart. Now he's saying that it's not working. What is a messenger RNA? Quickly. Anybody, what is a messenger RNA? What have you understood about it?
It's the processed form of the alien RNA which is formed by transcription from DNA.
Exactly. So that's what I wanted. So we have understood it's a long RNA. So how is it formed? So it's about only 5% in the RNA content of the cell. So what it does, how is it formed? Through transcription, it's formed. What is its major function? It brings the instruction from the DNA for the formation of a particular type of polypeptide chain. So that is messenger RNA. So now that means you know all about transcription. So what is the difference between replication and transcription? So both we did it in the previous class. So what is the difference between replication and transcription? Can you see the screen now?
Yes, ma'am.
Yeah. What's the difference between replication and transcription? So we are done with replication, we are done with transcription. How is it different? Come on, come on. Replication. Okay, let's discuss this. Everything if you write, then long time we need. So what's the difference between replication and transcription? So if this is also a CBSE question. I'm not asking anything out of the box or on my own. So I'm just asking these hot questions, differences between this, why tRNA is folded, what are the different loops in the tRNA, why these loops are formed. All these are hot questions. So if you know all these as good as NEET, you can, it becomes easy. So replication is what? Nothing but synthesis of another DNA. Transcription is what? Nothing but synthesis of RNA. From where? From DNA. From DNA, you are synthesizing RNA. So this is very, very important to know. Okay. In replication, both the strands take part in the process. Here, only one strand take part in the process. What do you call that? That strand? What do you call that strand which take part in the process in the formation of RNA?
Template strand.
Template strand. So there, replication produces double-stranded replicas of DNA. Here, single-stranded RNA is produced. Okay, so there, complete that double-stranded, that is duplex, DNA duplex unwinds, and both the strands separate throughout the length of the DNA molecule for the replication. For the Xerox copy to be formed, complete Xerox happens. Here, small part of the DNA unwinds. So for that, to make a template. So here, what else? There, okay, very important, another point. Primer is required. Here, primer is not required. Very, very important point. So what are the raw materials for replication and raw materials for transcription? Another question I have come across here, CBSE question. I'm looking into a CBSE question book. Okay. Hot questions. I'm looking into the CBSE hot questions from the CBSE biology handbook, CBSE handbook. Okay, so I'm asking, this is a very good question. What are the raw materials for replication and transcription? What answer are you going to write? 2 marks question. Very good question. What answer are you going to write? When these two are thorough only, then I can proceed with the next process here. What are the raw materials for replication and what are the raw materials for transcription? Guys,
Ma'am, are you talking about the enzymes or the nucleotides?
raw materials means how this raw materials for the formation of the double strand DNA. What is the raw material? So you need to produce double strand DNA. What is the raw material? What is required? Simple actually this is, but confusing.
Deoxyribose sugar and ATGC.
Yeah, so that is DATP for the formation of replication of DNA, DGTP, GTP, CTP, TTP, ATP. So which are D. So you have to add D because it's deoxy. Here, this is the simplest question, but appear to be very complex. Here, what is this is very confusing. If you are very confident, then only able to answer. Here, only a thing is in replication, you require DATP, DGTP, DCTP, DTTP. Here, you require simply ATP. Come on, tell me. For the formation of messenger RNA, what is required? ATP?
CTP, GTP, and UTP.
Yes, UTP, CTP, GTP, and UTP. That's all. So each quarter, and here also quarter. So it's a 2-mark question, simple question. Okay, so this is what they have asked in 2018 CBSE question. Nice question. Okay, now tell me the enzymes. Replication is catalyzed by replication is catalyzed by
DNA-dependent DNA polymerase and helicase, SSBP, topoisomerase.
correct. Which is a main enzyme?
DNA polymerase.
Polymerase, which one? In that also one is for proofreading. Which is the main? Polymerase 1, 2, 3 are there. Polymerase 3 in prokaryotes. In eukaryotes, there are so many. So polymerase epsilon, delta, all these are there. So now next is how the RNA is, which enzyme catalyzes RNA? Which enzyme catalyzes RNA? Can you tell me?
DNA-dependent RNA polymerase.
That is what? Why are you saying DNA-dependent RNA polymerase? What is the reason? Why are you saying DNA-dependent?
And because the transcription happens from the DNA strand.
Transcription happens?
And, it happens complementary to the DNA strand.
Yes. So suppose DNA-dependent RNA polymerase. So suppose if it happens in a reverse way, will it happen in a reverse way also? Do you think so? I want answer from other students also. Will it happen in a reverse way? Transcription? Yes. So formation of DNA from RNA happens. Will it happen? So we are coming to the point now. See, you said that, which is the enzyme? Once again, you tell me. Momita, once again you tell, so that let them all listen again. So which is the enzyme?
DNA-dependent RNA polymerase.
So which is formed from DNA to RNA. Okay. So now I'm asking, will the reverse would happen? So this is transcription, you said. That's right. This is transcription. DNA dependent RNA polymerase. So will there be the formation of DNA from RNA? Will it happen?
In the process of reverse transcription.
Reverse, yes. So why reverse transcription would occur? This is reverse transcription. Why would it occur? What's the reason for reverse transcription? Which is the enzyme now? Here. RNA dependent DNA polymerase. It is also called as reverse transcriptase. Why this happens? This happens only in viruses. So this is observed first time by Temin and Baltimore. So Temin and Baltimore observed this first time in raw sarcoma virus, RS virus or sarcoma virus. Okay, such virus which have RNA as the genetic material are called as retrovirus. So single-stranded RNA produces double-stranded DNA. So isn't this reverse of this? So this is nothing but reverse transcription. So the enzyme is reverse transcriptase. Okay, so this is discovered by Temin and Baltimore in sarcoma virus. Even COVID is a reverse transcription. So that's why RT-PCR. Okay, so we are done with this much. So now, when we are talking about after transcription, the messenger RNA, so we all discussed all about the cistron, muton, recon, so all these. So now, how this particular amino acid, so why do we call it the codes for amino acids? So that is comes under genetic code. Only genetic code part I would like to show the pictures and do it so it makes sense. So here is the genetic code. What is the genetic code? So this is nothing but the sequence of nucleotide. Okay. Just a minute. Just 1 minute. Just 1 minute. Where is it? Arya also has joined. Good. So why Arya a little late?
Mom, I came back from school.
Okay, so your mic is working, right? Arya.
Yes, mom.
Yes. So you cannot mute and say mic is not working, that is happening, this is happening. You cannot say no. Fine. So what is genetic code, Arya? What is genetic code? Code. Code
Mom,
means what?
genetic
Okay,
Ma'am,
what
it's a
is
three
code?
combinations of nucleotides which are called all that codons and all those things. Set of 3-letter combination of codons.
No, when I say, okay, how do you define genetic code? So the sequence of amino acids, right? So the relationship which forms the sequence of the amino acid on the messenger RNA or the nucleotide sequence. So what is that sequence? We are talking about the sequence of nucleotide in messenger RNA or DNA. So that is the in genetic code. That's called genetic code. So how this genetic code came up? So it is 3 bases determine a single amino acid, which is called codon. So why 3 bases only? Why not more? So let's understand here with respect to these scientists. George Gamow is a person who suggested that coding 20 amino acids, the code will be made up of 3 nucleotide, and therefore it is 4 to the power of 3, which is 64 codon. If it is single mono, so then it would be singlet code or single code, so then it would be 4 only. So but there are 20 amino acids. Which are those 20 types of amino acids involved in translation? So these are the 20 amino acids which you have learned in your biomolecules chapter. 20 important amino acids. If it is a doublet code, 4 to the power of 2, 4 4s are 16, then also it's not possible, insufficient. So therefore, 4 to the power of 3. So Gamow in 1954, actually, so he's a person who suggested the possibility of triplet code. So remember his name, George Gamow. So one codon consists of 3 nitrogen bases. Okay, so 64 codons are very much sufficient for all these 20 amino acids. So what are the contributions of other scientists given here? So you need to remember all these. So Gamow suggested triplet code. Hargobinda Khurana developed the chemical method in synthesizing RNA molecule with defined combinations of bases. So he created homopolymers or copolymers, whatever. Okay, so that's his contribution. And Marshall Nirenberg, what's his contribution? So cell-free system for protein synthesis, Marshall Nirenberg. Okay, and Okawa. So Severo Okawa. So what is his contribution? So polynucleotide phosphorylase. So that's enzyme which is used to polymerize RNA with a defined sequence in a template independent manner. So these are the contributions of these particular people here. Okay. So now what are the features of this? So this is how it is. So you can see the stop codon, and which are the amino acids which have only one codon. Which are those amino acids?
Methionine and tryptophan.
Methionine and tryptophan. What is methionine? What is the codon? You can
AUG.
see in this picture. Yeah.
AUG.
Yeah. So any polypeptide synthesis is always signaled by initiation codon. So which are those initiation codon? Usually, commonly by AUG, that is methionine, which is also initiation codon, and sometimes also very rarely by valine, which is GUG. So where is valine? So it is a GUG, start codon. There is one more start codon, that is GUG. Okay, so you can mark those two. AUG and GUG. So GUG, where is it? Here. Sometimes, but always methionine. So what are those which are given here as stop, 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 stop codon, stop signal. So it is a termination of the polypeptide chain. Whenever this codon comes, that means stop. So whenever red light comes, it stop. Similarly, whenever green light comes, it start. So this is starting. So these are stopping. So which are those codons? Which are those? UAA, UAG, and UGA. So UAA are called ochre, given particular color, and UAG is amber, and UGA is opal. So that means they do not specify any amino acid. That's the meaning of it. And hence they are also called as nonsense codons. No sense. Nonsense codons. Okay. So codons are universal. Why do you say they are universal? So what are the features, salient features of the genetic code? Is a common question asked, easy question, scoring question. Okay, scoring answers you can always score. So codons are always in triplet. 61 codons, because 3 are non-stop, are code for different amino acids, 20 different amino acids. Okay, so because they are a termination codon, so that is nonsense codon. They do not code for any amino acid, hence they stop the polypeptide chain growth. So AUG has dual function. What is that? It codes for methionine, also an initiator codon. In eukaryotes, it's the first amino acid to be attached, and formal methionine in prokaryotes, F-methionine, F-met. So an amino acid is coded by many codons. Okay, and AUG for methionine and UGG for tryptophan. Only these two have one single codon. And why do you call codons degenerate? Why do you call them such codons degenerate? What is the meaning of it? Degenerate. Anybody? What is the meaning of it? Degeneracy. What is the meaning of degeneracy of codon?
A
Yes.
single amino acid is coded by multiple codons.
Yeah. So why do you call it degeneracy? All other amino acids are specified by 2 or more, isn't it? 2 to 6, basically. So when they are coded by 2 to 6 different codons, so they are redundant. So this is called as degeneracy of codon, redundant. So in that regard, we have Ubel hypothesis. So have you, is it given in your textbook, Ubel hypothesis? What is that Ubel hypothesis? What it says? Degeneracy of codon. So this is discovered, incidentally it is discovered by Nirenberg and, please write it down. It's discovered by Nirenberg and Bonfield. Okay, so this is the degeneracy. So now I want you people write the different codons for different amino acids, leucine, serine, arginine. So which are the different amino acids they code for? Okay. So what is this Ubel hypothesis? This is Ubel hypothesis is also discovered, that's told by Crick. Why it Ubels? What is the Ubel hypothesis? See, anticodon normally recognizes only one codon. Sometimes this anticodon recognizes more than one codon. So that means it's wobbles, it's wobbling. So wobbling is the, what is the meaning of wobbling? What is the meaning of wobbling? Crick told this Ubel hypothesis. Did I put here? Ubel hypothesis. Yeah, white vowels. So anybody? So third nitrogen base has no effect on coding, and the same is vowel hypothesis. So that means here AG can recognize two codons, that is U and UC. Go to this and see here. So here, so both stand for phenylalanine. So this is, so it vowels because of many 2 to 6 codons. So that is the meaning. So codons has no punctuation between the adjacent codons, and it's read in mRNA in a continuous fashion. So genetic code is universal. So from bacteria to human, the same triplet code for the same amino acid. So some exceptions are found in mitochondrial codons and in some protons. Codons are non-overlapping. What is the meaning of it? Non-overlapping. Non-overlapping, non-unambiguous. What is the meaning of it? Unambiguous means what? No confusion. That particular codon overlap for that only. So nitrogen base in a particular codon is a constituent of only one codon. So that is the meaning of non-overlapping. So particular nitrogen base is a constituent of that particular codon. Okay, so that is comma-less, it's shown in the picture. Universal, that means it's the same in all the organisms. And non-overlapping, that means the codon, whatever the nitrogen base, it is there. So it codes for that particular amino acid only. And unambiguous, so they, because of that non-overlapping, there is no confusion and specificity is there. So you need to know these things. Okay, so how this mutation and genetic code are interrelated? So what is the relation between genes and DNA? So they are best understood by mutational theory. So deletions, rearrangement in a DNA. So all these would cause loss or gain of the DNA, and so therefore its function. Insertion or deletion of one or more bases change the reading frame from the point of insertion or deletion. So you all know about it. So anything which you insert or delete, it changes the master copy. Okay, so that is the meaning here. So we already learned this frame shift mutation in the previous chapter. So you know about it. So insertion, deletion. So what are the different mutation? Fatafat, can you tell me? Can you recall? Arya, can you recall? Arya, can you recall different point mutation, frame shift mutation? So then based on the number, which are the different types of mutation? Based on the structure of chromosome, what are the different types of mutation? Insertion, deletion, inversion, transition. Okay. Ploidy, with respect to ploidy also it changes with respect to number. Then you have, which are the different type? Trisomy, monosomy, nullisomy. So aneuploidy, euploidy. Under that you have studied all these. So let's go to now. So this much is the information with respect to genetic code. So you know now what is genetic code. Let's go to translation now. Okay, so let's go to translation. What is translation? Anybody? What is translation? Anybody?
the codons and the mRNA are read and the corresponding amino acid chain is formed in the ribosome.
1 minute, just a minute. Yeah, what is the? Okay, translation. So what is translation, students? And that's what I'm asking. What is translation? What's the meaning of translation? Arya? What is translation, students? French is translated to English. So why do we call it translation? Yeah. So protein is formed. So what is translated? The code present in the messenger RNA is translated to protein. So the process of making protein. So what is translation? The process of making protein, that is a polypeptide chain. Polypeptide chain is a protein. Using the genetic information, carried in messenger RNA is translation. Is that clear? So that is translation. So how this translation occurs, let's understand in detail. So to understand translation, what are the substances that is required? What are the machinery? What are the components of translation? Can you tell me what are the components required? So now we have come to the final thing. So we have made everything and now we have to translate. So what all we have made, we have understood. Come on, one by one, go on giving. So everybody should answer now. So what are the different components required for translation or machinery for translation? So messenger RNA is the template, right? So very first, let's write each of them, machinery for translate. So we need ribosomes, so that you will understand what are the points. So then we will understand the how to make it. So like how to understand a recipe, ingredients, and then process. So ribosomes. So ribosomes are protein factories, we know. So when we say ribosomes, ribosomes have two parts, smaller unit and larger subunit. So this larger subunit of ribosomes has a groove. Okay, so the larger subunit, so smaller subunit and larger subunit here. Okay, so this larger subunit has a small groove, and this groove is for pushing out the newly formed polypeptide and protect them from the other enzymes. So here, when they have to prepare the protein, both the subunit, that is smaller subunit and larger subunit, unite together, come together. So how are they formed? So the two subunit come only at the protein formation, otherwise they are separated and present in the everywhere. This phenomenon of coming together of the two subunit is called as the first association of ribosomes. So what is required for this association? Magnesium is required for this association need point. Okay, so magnesium is required for this association. So after the completion of protein synthesis, again they separate. And when they separate, it is dissociation, as you know. Okay, so this much is the information you should know with respect to ribosomes. Okay, so different ribosomes, when they are held together by the messenger RNA, what do you call? So this is the messenger RNA. So this messenger RNA hold different ribosomes. What do you call those different ribosomes? What do you call these different ribosomes? When they hold together like this, they hold. What do you call that? A strand of messenger RNA holding different ribosomes. This they are called as polysome or polyribosomes or polysome. So what do they? They help to produce the number of copies of this same polypeptide. So polysomes. This is. Now, so different parts of this ribosomes, let's understand, which connect with the protein part. So very first is a channel for messenger RNA, which is between the two subunit you can see. So that is the channel for ribosome. So very first is channel for messenger RNA between the two units. Okay, I'm making it very in a simple language, not a textual form, so that you understand this translation easily. So after this channel, so then there should be an exit tunnel also. So why? So this exit tunnel helps to the movement of this newly synthesized pop from the larger unit. So larger subunit will have exit tunnel. So larger subunit will have exit tunnel. For example, so let me show it in a picture so you will understand that better. Suppose this is the messenger RNA, this is the smaller subunit and this is the larger subunit. So here you can see for the attachment of messenger RNA, there will be a groove. Here you will get to see the messenger RNA. So let me write messenger RNA here. So messenger RNA is a particular codon and then anticodon. So this will be the codon for messenger RNA. Messenger RNA. And here there is a So here there is an anticodon on this loop. So this is tRNA with anticodon. Okay, so now as they go on building, so now this will be the exit tunnel. So this will be the exit tunnel in this messenger RNA, in this, sorry, larger subunit. So the exit tunnel will be like this, from which the different polypeptide chain will grow during translation and exit. So that's all is the actual fund up here. So they exit out. So when they exit out, they get folded and form respective protein. So this is the 60S subunit and this is that 40S subunit. Is that clear? So this is messenger RNA with codon and this is tRNA with anticodon. This is how it happens. Where it goes after formation? Okay, let's do that later. So how they are formed? So they start coiling like this, tertiary structure, then whatever. Okay, but this is the primary structure, then they start forming secondary structure, then it becomes the tertiary structure and whatever. So now, what is the third point here? You need to know. So here, channel is there, exit tunnel is there. Point number 3 is, so they have 3 reactive site. These have. So which are the 3 reactive site? 3 reactive sites are there. These sites are P, A, and E. What do you mean by P and E? Can you tell me? P site for? What are those P site? Peptidal transfer. So for the transfer of peptide, we are talking about the structure, not the process yet. So this is peptidal transfer. Peptidal transfer. What is A site for? Amino acyl site. A is amino acyl site. And what is E site? What is E site? E stand for exit site. Okay, larger subunit. So like how I showed you, this is the exit site. Here, exit. Exit tunnel, this one, and the site is here. So that is exit site. So these are the 3 parts of this ribosomes. So now, when we say peptidal transferase, what is that? So it's a component of larger subunit and it is 23S rRNA in prokaryotes. So here we are talking about prokaryotes, so therefore it is 23 rRNA. So this peptidal transferase, very important, hot question, hot NEET question. This peptidal transferase is a ribozyme. Is ribozyme. So smaller unit has a point of for recognizing messenger RNA and binding area for initiation. What are the points on this smaller unit? So all these we are finding it on larger unit. Okay. So larger subunit. So what is there for smaller subunit? So it has binding site area for initiation factor. So smaller subunit has binding site for initiation factor. There are different initiation factor, 3 in prokaryotes, 9 in eukaryotes. So these are initiation factors. So which initiates translation. Okay. I have started here. Okay. Shall I rub this? Is it required for you? Guys, shall I rub this?
Yes, ma'am.
Because hopefully you have taken down that. So let me rub that and okay. So what is the next machinery? The next machinery is amino acids. Amino acids also required. So there are so many different types of proteins we have in our body, in organism's body, in a single cell. So all these proteins are formed from the 20 amino acids in our body. So where are they present? So amino acids are present. 20 amino acids are present in the cellular pool. So all the biomolecules present in the cell is a cellular pool. So they are present in that pool. So the next component required here is messenger RNA. So we all know that. So ribosomes over, amino acids over, now messenger RNA. So where is this messenger RNA? So messenger RNA bring the coded information. Coded information from DNA. So that is where DNA is responsible for gene expression or protein formation. And this take part in translation. So by bringing that particular amino acid in a particular sequence to form a particular polypeptide. So now the codons of this messenger RNA are not recognized by amino acid, but by anticodons now. So where are these anticodons? So these codons are not recognized by amino acids, but by anticodons. Where are these anticodons? Where are these anticodons? Amrita. So anticodons of the respective adapter molecule. I'm giving you the clue. Amrita, where are these anticodons? See, the codons of this messenger RNA are not recognized by amino acid, but it is recognized by anticodons to attach that particular amino acid. So where are these anticodons? My question. Anticodon loop, where are they? They are on tRNA. Are you understanding? So tRNA activation is required. We will talk about that. So which tRNA required? So tRNA should get activated. So when they get activated, we call it amino acyl tRNA. So what is that amino acyl? Let's talk about that. So these have anticodons and these help in transferring the so they pick up this respective amino acid and help in getting it transferred here to, or else to help in getting the polypeptide chain grow. That you can tell. So let's have an overlook of this translation in an animation. So what is the last one? Okay, let me write the last component. Last component is this amino acyl tRNA synthetase to produce amino acyl tRNA. So amino acyl tRNA synthesis is required. So for that, synthetase is the enzyme. So these are the components required. So this is an enzyme which helps in adding amino acid to its particular RNA. So this is also called as amino acid activating enzyme. So activation of tRNA is required very first. So activation of amino acid, activation of this amino acid, then it will get bind to this charged tRNA. So charging of tRNA with the specific amino acid. So amino acyl synthetase activates amino acid, and this is charged tRNA. So these two are very important. Now, to understand all these, let us watch a 2-minute video or 1.47, some animation. It's a beautiful video from McGraw-Hill. So let us watch that and then understand the complete concept in detail. So if you watch that, it becomes very easy for you to understand. This is a very nice video. So I felt you should watch it. So I thought I'll show it. Just a minute. Yeah. Just tell me, can you see the screen? Do you get to see and hear?
No, ma'am.
Guys?
No, ma'am.
Oh. Please tell me now.
We need to supply them with
Now?
enzymes to break down the food
Yes, ma'am.
particles into
Yeah.
usable nutrients our cells can
No,
absorb.
this, okay.
And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is
You are getting to hear also, right?
Yes, ma'am.
Okay.
made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
So you have got a very good explanation, animation. So overall, it's a summary like. So now let's understand the mechanism of this translation. So how the mechanism of translation occurs. So we have understood the components. So now let's go to the mechanism of translation. So very first is activation of amino acids. So it's carried out by activating enzymes. So it's carried out by activating enzymes, and these activating enzymes are called as amino acyl tRNA synthetase. Amino acyl tRNA synthetase. This is given by Hogland. So what is this activation means? So here amino acid, basically let me depict it in the form of an equation. Amino acid in the presence of ATP and the amino acid synthetase enzyme, this enzyme. So with the magnesium as a cofactor. So what it becomes? So it becomes amino acid. So ATP is here used for the process. So the energy is available for the formation of this activation. So it produces amino acyl adenylate enzyme complex. So amino acyl, this becomes AMP and enzyme complex. So here inorganic phosphate, so is removed. So pyrophosphate is removed. Is that clear? So this enzyme is tRNA synthetase. So hydrolysis of this pyrophosphate with the help of pyrophosphatase produce energy for the next initiation reactions always. So what is this? This is the first step. Step 2. So let's do stepwise, then it becomes easy. So charging of tRNA. So amino acylation of tRNA happens, or it's also called as charging. Charging of transfer RNA. How it happens here? So as we know, charging, so specific activated amino acid is recognized by specific tRNA, we know that. So now this amino acid attaches to the amino acid attachment site. So we know that A site is there. So on this ribosome, so amino acid attach. Where is amino acid attachment site in tRNA? Where? Which end?
3
Right,
dash.
where? 3' exactly, 3' OHN, 3' hydroxyl end of tRNA. Okay, so this is charging of tRNA. So here enzyme is also there, enzyme is released, here enzyme is released, AMP is also released. So now this amino acid, okay, so will get attached. So this is linked to, so this is attachment of amino acid to 3' OH end of tRNA. So how it attaches? Through its COOH group. Through its COOH group. Are you understanding? Oh, what happened? The screen sharing is gone, is it, for it now?
Yes, ma'am.
Oh my God. see here. Can you see now?
Yes, sir.
Okay, so if we have to depict one equation kind here, so then how we can do amino acid. So AMP is attached with enzyme plus tRNA. What do you get? So you get amino acid tRNA. And this is released. Enzyme is also released. Okay, so next is initiation. What happens next? After charging, initiation occurs. Initiation of the polypeptide to group. So initiation requires initiation factors. So in prokaryotes, there are 3 initiation factors. Initiation factor 1, initiation factor 2, and initiation factor 3. Whereas in eukaryotes, we have been learning prokaryotes as it is a simpler one. So in eukaryotes, there are many initiation factors. So eukaryote initiation factor 1, eukaryote initiation factor 2, eukaryote initiation factor 3. Okay, so eukaryote initiation factor 4A, eukaryote initiation factor 4B, eukaryote initiation factor 4C, 4D. So these are different factors. Factors are also proteins, of course. So they are also formed by this process only. So initiation factor 6. So here 2 and 3 are attached. Okay. So all these are the factors. So here, out of this, 2 and 3 are attached to the smaller subunit of ribosomes in the dissolved state. So here, for energy formation, what can you see, students? Is it shown?
No, ma'am.
I'm sharing. I'm sharing. I'm getting the notification. I'm sharing screen.
Yes, ma'am, it's visible now.
Oh, maybe the internet is okay. So here, for the attachment of the factors, what is required? GTP is required. So this messenger RNA, now what is the next step here for initiation? Messenger RNA attaches itself actually to the smaller subunit of ribosomes as we discussed. where it attaches to the methylated. Okay, so to that methylated cap region, cap and tail are there to the cap region. So what is that cap region? So this cap region, so here let's write messenger RNA attaches now. Messenger RNA attaches itself to smaller subunit of ribosomes. At cap region. So this cap region has nucleotides complementary to the nucleotides. Okay, come on, tell me this cap region, what the nucleotides are complementary to which one? Messenger RNA cap region has the nucleotides complementary to, so because of the attachment, so you can figure out. Come on, tell me, this is the cap region. So this cap region, just now you've looked into the animation also. So this is the cap region. So now this cap region has the nucleotides. So when smaller subunit attaches to it. So come on, tell me. This will have the complementary, so of the ribosomal RNA also present at the 3' end. Please understand. So this attachment is such that the initiation codon of messenger RNA, that is AUG or GUG. So cap region where ribosome RNA will have the complementary, so you can write that, where I'm making it very in a simple language instead of textual, because children think that this translation is very difficult, as if I have experienced. So this is an easy one actually. So messenger RNA attached to the smaller subunit of ribosomes, and which has the complementary nucleotides. Complementary nucleotides at that 3' end. So which is the codon at messenger RNA now? So here the codon is, which is a codon? Arya. Here the codon of messenger RNA is which one? Arya. Arya? For the initiation to happen, what is the codon? Arya is there, right? Arya, you are there? Yes, I can see you. Come on. Here the codon of messenger RNA is.
Here is a cell, the basic
God, what
unit
is
of
this?
all living tissue. In most human cells, there is a structure called the
What is this? Arya, I'm asking you, which is the codon present there on messenger RNA? Anybody? Here the codon of messenger RNA is
AUG.
AUG. Always, most of the time. Sometimes, what is the other possibility? Is it always AUG? All the time? Predominantly, always, most of the time. Or
GUG.
GUG. So the site is which site? Site. What do you call that site? APE. Which site?
P.
With P site. P is for at P site. P is for
Peptidal transfer site.
peptidal transfer site. Okay, so this is how it happens. So to write the reaction, once again, let's write the reaction for all of that. So 40S subunit with messenger RNA. So with the initiation factor 2 in eukaryotes, or 3. Okay, let's not write eukaryotes. So initiation factor 2 and 3. Here we are talking with. So initiation factor 2 or 3. So now becomes 40S messenger RNA. So that means smaller unit attaches. Okay, so it could be methionine or valine. So this region. Now what happens after, soon after this, I am writing it just like one by one, stepwise. So now soon after this small rRNA attaches to that site. So what is that going to happen here? Amino acyl tRNA complex. Amino acyl tRNA complex. Which is this tRNA complex? So it is specific for a particular codon, initiation codon, that is methionine or valine, whatever. So reaches this site. Now reaches this P site. What it will have? It will have the anticodon. Okay. So which is that anticodon? Here. Anticodon, it is UAC. Anticodon. AUG means A instead of T here, and this is C and this is C. So with the anticodon, UAC reaches there. Okay. And establishes. It establishes what? It establishes temporary hydrogen bond with the nucleotide there, with that initiation codon in the messenger RNA. So now we have codon anticodon reaction occurring here. Okay, so this complete is energy for this particular reaction is provided by GTP, as I told you. So now let's write that depiction. So mRNA, you can write only the depiction for your CBSE. That's why I'm showing this. So plus tRNA. tRNA is methionine. So tRNA met, formilated methionine, F met. So now in the presence of initiation factor 3 will help to attach here. In the presence of with the GTP, what happens? So it forms 40S messenger RNA and tRNA complex with MET. So this is how it happens. Is that clear? So this is what happens in this particular thing. So now what is the next thing happening here?
Ma'am, is this for prokaryotes or eukaryotes?
So here we are talking with respect to prokaryotes. Why? So in prokaryotes only F1, F2, F3. So in eukaryotes, it's a very complex point. So very important point here. One important point you need to remember here is, so what is that? We are talking with respect to prokaryotes. So in prokaryotes, there is a sequence called Shine-Delgado sequence, SD sequence. So here in prokaryotes, there is a sequence, SD sequence, named after the person, that is Shine-Delgado Delgado sequence on messenger RNA which recognizes the, let's write, recognizes the smaller subunit
Yeah, but in a prokaryote, won't the smaller subunit be 30S and not 40S?
Okay, so here, okay, I have depicted it as 30. Yes, you're right. Here we are depiction, so you can write it as E here. So E is for eukaryotic factors, 2. So here mostly specifically 2. So because I am showing the depiction, so you can write it as 30 also here. So this is one hot questions that can be asked in NEET. So this is a high-end question. So this is, I'm putting that in the box. So if you want to show it for eukaryotic, so you can write it here. So what is that? 30 subunit less messenger RNA in the presence of IF3 and magnesium factor. So what happens? So 30 subunit messenger RNA complex. So this reacts with formilated methionine tRNA complex to form messenger RNA, formilated methionine tRNA complex to form that particular. So here we can write it as, again, 30S messenger RNA, the same thing. Messenger RNA complex plus FMET tRNA, FMET, you can write it on the surface also. So in the presence of GTP, with the help of GTP, IF2 and 3. So with the magnesium as a cofactor, so you get 30S messenger RNA, tRNA FMET. Okay, complex. So now it is ready. Now this is ready. So this is all about the initiation. Now what happens here? As I told you, the larger subunit has P site, A site, E site. So now what is the next thing happens here? The next is elongation. Elongation takes place. Chain elongation. So now the new tRNA. So now the new tRNA. So with new amino acid, new tRNA, new amino acid, where it attaches? Attaches to which site? Which site, Amrita? Amino acid attaches to A site of ribosomes, larger subunit, A site. So how this link between the amino acid of P site of tRNA? So what happens now? The moment it attaches, remember, recall the video, recall the animation. The moment it attaches to A site, what happens? What happens? What happens the moment it attaches? Amino acid at A site. Amino acid at, P site, that is peptidyl transferase site of tRNA is broken. So it breaks. So it's broken. And what happens? It's broken. And what happens to that? What happens to that?
It makes a peptide bond with the next amino acid.
Correct. So to show to depict that. So how can we depict? Let me try to do it in a. Okay. So let me show this as the messenger RNA. It is not shown. You can depict also in your CBSE exam. You will get marks, of course. So this is that. So this is AUG. Next. So only these things you AUG. That's all you do. So what happens to this AUG? Smaller subunit is attached. Smaller subunit. Okay. So this smaller subunit. So now this is the larger subunit. And this larger subunit has A site, P site, E site. So now this is the A site. This is the P site. So now with this P site, you can show this tRNA attachment here. Okay, so tRNA with the first amino acid. So first amino acid, I am showing it with another color. Okay, this is the first amino acid. One I will write here. One. So this is there just like that here. This side. So what happens next? So second one will come and sit here. So when second one will come and sit, so you have to show now. Okay, let me, because there is no space, I'm going to do it here. So second one will come here. So what happens to this? This is the larger subunit. So for this larger subunit, so what should I write? Is it 5 dash or 3 dash this side? What is the end here? Here. What end is this? That's very important. What end is that?
5 dash.
5 dash. So this end is 5 dash and this end is 3 dash. So it's understood. So now it's not looking good. Wait. I'll do it little bit this side. So for your understanding, I'm just doing. So let me draw this. Okay. So what happens next? So this is the, and here is the larger subunit. So this is your A site. And this is your P site and this is your E site. Okay. So what happens to the A site? What happens now? Peptide bond, the link between the moment tRNA attaches to the A site. So now the new tRNA, so that is here, it attaches. So this is the another amino acid. So let's call it 2. So the moment it happens, so now it breaks and one will come here and 2 will be there. So now another will come. So what happens here? 1, 2, and 3 will come here, attaches here, the next step. So like that it grows. Okay. Are you understanding? So this is what will happen here. So the bond, the amino acid at that P site breaks of tRNA and tRNA of P site is discharged. So how this is formed? Now peptide bond takes place between the, so what happens now? Point number 3. So now peptide bond between COOH group of P site amino acid how this form this bond. So P site amino acid and amine group of A site amino acid. Peptide bond is formed. So here NEET point is 23 srRNA. This rRNA initiates, induces, or triggers the formation of, induces the formation of peptide bond. So this is very important. What do you call that RNA which acts as an enzyme? What name is given to that rRNA which acts as enzyme? Biomolecules also it is there. What do you call it? Ribozyme. So this is called ribozyme. Any rRNA. So which rRNA acts as ribozyme during protein synthesis is 23 srRNA, which is an important point. Please remember always. So very, very crucial NEET question. Okay. So this induces the peptide bond, and this particular rRNA is called as ribozyme. So ribozymes are ribosomes. rRNA which act as, RNA which act as enzymes. Now, finally, what happens here? Finally, so let's come to the termination next. So what is the next termination? So let's completely understand. So peptide bond is formed. So once peptide bond is formed, what happens next? So the attachment happens. So now, after that attachment, what happens next? Quickly. What happens next? Formation of peptide bond. So now all these, this now tRNA of A site is transferred to P site. So now A site is empty again. So again new one will come. So now ribosome slides over messenger RNA like this from 5' to 3' end. So here we can write this way. So this way ribosomes keep sliding, slides over messenger RNA. How? From 5' to 3' direction. So due to the sliding of these ribosomes on messenger RNA, so what is happening? New codon of messenger RNA is continuously available at A site of ribosomes. So based on that new code, whatever is there, new amino acid is go on getting attached. So here another important NEET point is, so translocase enzyme. Translocase is the enzyme which helps in the movement of ribosomes. And GTP already we have shown here. GTP is also an important point. So, and translocation happens because of translocase enzyme. Helps in movement of ribosomes. Okay, finally how it looks. So what happens finally in that terminations? So now elongation. So elongation is the attachment of this amino acids one by one, and it forms elongation. So how this elongation occurs? Elongation, initiation for initiation factor and elongation by elongation factors. So now let's call the elongation. So here, elongation occurs due to elongation factors. Due to elongation factors. So in prokaryotes, so 3 elongation factors are present. So which are they? Which are they? Anything is given in your textbook? And any such elongation factors are given in your textbook? Three elongation factors are given. So the most abundant in prokaryote, which is found out in Escherichia coli, is, so that is elongation factor TU. Okay, so this is named after the particular enzyme here. So elongation factor TU is very commonly occurring one. Okay, so after elongation, what happens? Termination. So termination means stop. So how this termination occurs? Termination occurs due to the nonsense codon or stop codon. Which are those stop codon? So whenever, as these ribosomes go on sliding, so now let's say they come across any of the codons. Which are those codons? Termination can occur because of any 3 codons. Which are they at A site? Which are those? Guys, Anish. Anish is not there here. Vihan, can you tell me which are those codons? So now this is A site. So here in this A site, we come across this. Anybody? Arya, which are those codons? So P, A. So when they come across, guys, can you hear me?
Yes, ma'am.
Okay. So UAA. Is it UAA only? Which are the 3? UAA, UAG, UGA. So whenever any of these comes, so then it stops. So by now here, what has happened? So here in this tRNA, so there will be synthesis of so many polypeptide. So this polypeptide chain exit through the exit tunnel here. So it exits through the exit tunnel, as I told you. So once they come out, you know. So here, how they release for the exit of this. So after elongation. So now termination. So what is termination? Termination is brought by whenever nonsense codon. I have written already there. So available at A site. At A site, then polypeptide chain terminates. Now the polypeptide chain is broken. So now the polypeptide chain is broken. By release factor. release factor, initiation factor, elongation factor, release factor. Even this release factor are 3 in number. Release factor 1, release factor 2, release factor 3. So these are the 3 release factors. So release factor, release this. So for all these, GTP is required. Keep in mind, everywhere GTP is required. So this messenger RNA, okay, is released through that. And what happens? What is the fate of this messenger RNA? Now tell me. So what is the what is the fate of this polypeptide chain? What happens? What is the fate of this? Guys, I'm asking you, what is the fate of this released polypeptide chain? So you saw that in the animation video. What happens to that? Varshini, please tell me what happens to the, this is the CBSE question. What is the fate of the polypeptide chain formed during translation? What answer are you going to write? Arya, can you tell me, dear, what is the fate of this polypeptide chain? Arya, I'm asking you. So here throughout the protein synthesis, lot of proteins. Momita?
When it exits the larger subunit through the exit tunnel, then it forms its secondary or tertiary structure, and then it goes through the endoplasmic reticulum and the Golgi body.
Correct. So that is next is modification. So that's called as modification. Modification of this. So what is modification? So now the polypeptide raw material is formed. So now the raw material has to get modified to perform different functions in different places in our body, in organism's body. Okay. See, this is the primary structure of the protein. So now it has to get coiled, folded to secondary and tertiary to be functional. So it will get associated with other polypeptides also, which are formed earlier. So different, that is, foldings would occur here. So for that foldings to happen, where that processes, PMP occurs, where PMP occurs, processing, modifying, packing. So processing and modifying in endoplasmic reticulum and packing in Golgi apparatus. Okay, so these proteins, some enter into the endoplasmic reticulum and some directly into the Golgi apparatus. So here they get, okay, so modification occurs in what we can write it as okay, so here these proteins are released into lumen of ER in cisternae and tubules, basically. Okay, lumen of ER, then to Golgi bodies for processing, modifying and packing. PMP, remember, packing. So that's why they are called as movers and packers. So that is called as, what do you call the modification of protein? This is called as, what do you call the modification of lipids? Glycosidation, and here it is called as glycosylation. glycosylation. Glycosidation is for this. So modification happens by the addition of sugar. So when sugar is added for different purposes, modification, that's called as glycosylation. Addition of sugar to that protein. So these are packed into vesicles, and these vesicles are released from that. Okay, one such vesicle is lysosome only. You all know about it. Okay, so now, very important point, one point what I want to tell you here is, so this is also a NEET point, little high-end NEET point. So I'm going to do it red. Red means little high-end, pink means NEET point. Remember like that. So when there is a formation of number of copies of the same polypeptide chain from that RNA, from one RNA. So with the help of this polysomes means so many RNA. So this is called as messenger RNA with so many ribosomes. This is called as translational amplification. So what is translational amplification? Translational amplification is nothing but formation of number of copies of of same polypeptide. Simultaneously. That is very important. That is why amplification it's called as. Simultaneously, that means at the same time from messenger RNA. So that is polysomes. That's why in many messenger RNA is called as translational amplification. So this much happens. So now this particular, so in prokaryotes, whenever a person takes some antibiotics, so what happens to the translation? So some of the antibiotics are seriously affect the translation in prokaryotes. So that is bacterial protein synthesis. That's why we take these antibiotics. Some may rupture the plasma membrane, some will stop the protein synthesis, and therefore the metabolic activities or the biochemical pathways in the bacteria is stopped, and that's how it's possible for us to eliminate bacteria from our body. That's why always you have to take a course of these antibodies. So some of these antibodies we can cite here are streptomycin. You all have understood, I mean, all have heard streptomycin, what it does. Streptomycin inhibits translation, and when it inhibits translation, misreading takes place. So streptomycin is one. Next, tetracycline, another antibody. What it does, so it inhibits, this inhibits translation overall. Broad spectrum antibiotic. Sometimes you can take it for general thing, also general infections. So this inhibits binding of amino acid tRNA to ribosomes. So what chloramphenicol? What this does, this inhibits peptidyl transferase, that enzyme. Peptidyl transferase. This enzyme is inhibited, so further formation would not happen. So like that, there are so many. So neomycin is another one. Neomycin, what it does? So it inhibits interaction of tRNA and rRNA. So, sorry, mRNA. So inhibits interaction. This is very important for the formation of peptide bond. So interaction of messenger RNA and tRNA. So that only does not take place here. Okay. And what else? So these are some of the points you can write down. So erythromycin is also there. Erythromycin is another antibiotic you all would have heard. This actually inhibits translocation of ribosome from along, inhibits translocation of ribosomes. So ribosomes has to get translocated along messenger RNA. That only does not happen. So therefore, in all these cases, one or the other does not happen means finally peptidal bond would not take place. So that's what will happen here. So this much is all about the translation. So I want you people give me the difference between transcription and translation, and then we can move ahead. So one of you please give me the differences between translation and transcription. Arya, over to Arya. I want Arya to give this. Arya, can you give me the difference between translocation and transcription? Fatafat. Arya? Okay. Arya, are you there? Come on, Arya, give me the difference. Transcription means what? Transcription is a formation of RNA, messenger RNA from DNA. What is translation? Formation of polypeptide. Okay, all of you give me one difference. That would be better. Vihan?
Ma'am, can you say the product form?
What do you mean by that?
Ma'am, product form in transcription, it is mRNA and all that. Other one is protein.
Correct. So translation is polypeptide chain form. So what is transcription? Which one act as template? Which DNA strand act? What do you call the DNA which act as template? Antisense. And here the template is messenger RNA. So because based on that codon, anticodon is there. So where it occurs transcription? In the nucleus. Translation, you know everything. Now you just go on bringing the differences. So this is an application-based question. Such kind of questions only will be asked in CBSE. So here it occurs inside nucleus, and in translation it occurs in cytoplasm. This is another important point. But where transcription occurs in prokaryotes, is there nucleus? No. So therefore, transcription and translation occurs in the same site, that is cytoplasm in prokaryotes. This is an important point, highlighting point. Please keep in memory. So what are the raw materials for transcription? ATP, GTP, CTP, UTP, we already discussed this. So here the raw material are amino acids, which are 20 in number. So that is another important point. Okay, so transcription helps in the formation of messenger RNA alone. Is it only messenger RNA, Momita?
Ma'am, mRNA, tRNA, and rRNA.
Correct. So here, translation.
The end product is a polypeptide.
Polypeptide chain, and all these RNAs are used here. Okay, so the enzyme, now coming to the enzyme, transcription requires RNA polymerase. Here translation requires different factors: initiation factor, elongation factor, termination factor and translocase. So next, releasing factor, sorry, not termination factor. So next, so that much only, I think, right? So this much is the difference. So here all, okay. Lastly, the modification. What is the modification in transcription? Capping and tailing, right? Is it only capping and tailing? Capping, tailing of messenger RNA and splicing, spliceosome splices the introns and ligase attaches the exons. So here, what is the processing here? Amino acid processing, tRNA charging, and also requires once the polypeptide chain is formed, what happens to the polypeptide chain? Modification happens, and modification when sugar molecules attach, it's called as glycosylation. So what it, and then it is packed into the vesicular forms. So vesicles, one such vesicle is lysosome. So this much is all about this. And take a break. What's the time now? Take a break for 10 minutes. It should be enough. So then we will come back and learn operon concept or regulation of gene expression. So all these we will understand now here. So take a break. Okay, so now it is 7:18, 12 minutes break. Take a break, come back at 7:30. So we shall continue. Yes. Guys, are you there? Vihan, Momita, Amrita, Arya. Are you all there? I want attendance quickly. Hardly 4 students and no attendance. Why? What happened to Anish? Anyone is Anish's classmate? Anybody is Anish's classmate?
Yes, ma'am.
Okay. So what happened to Anish? Why is he not coming?
Ma'am, I'm not sure. I don't know.
Oh, okay. Okay. So now we thought of understanding gene expression. How is it? Expression of the gene is regulated by what mechanism? What is that? So it's a particular mechanism at the molecular level. So where the gene is able to express itself phenotypically in the, where the gene is able to express the phenotype of the organism. That's a better way. So what it involves? So this is nothing but biochemical genetics. Why? So because it consists of synthesis of specific RNA, specific polypeptidopeptide, and structural gene, proteinaceous enzymes, all these are involved. See, actually, there are 2 types of genes, housekeeping genes and regulating genes. So housekeeping means what? Which are continuously expressing themselves in all the cells of the body. So because their product is always required. So for example, glycolysis. So whatever the enzymes required for glycolysis, it's always required. So it is their housekeeping genes. They're also called as constitutive in many of the books. They also give it as constitutive genes. Okay. So other genes are there. You want me to write these? So I'm giving the introduction to the gene expression. You want me to write? Guys, I'm asking you.
Yes, ma'am.
Arya, are you there? Vihan? Beta, are you there? Vihan? Vihan? Amrita? Amrita Varshini, are you there?
Yes, ma'am.
Okay. So there are 2 types of genes I was telling you. So what do you call one? Constitutive gene and another non-constitutive gene. So what do you mean by constitutive gene? Anybody? Constitutive, as the name says. Am I audible?
Yes, ma'am.
Okay, so they are housekeeping. So housekeeping means required all the
Are you
time.
writing something?
Yeah. Can you see the screen, guys?
Mom, the screen is frozen.
No, others can see?
Yes, ma'am.
Yeah, how come? Arya?
Mom, now it came.
Okay. So all those genes which are not expressing their effect all the time are non-constitutive. So their activity is regulated. Okay, and therefore they are regulated gene. So how do you understand this? So therefore, gene regulation. So how we can put it as? So gene regulation is the mechanism of switching off or switching on. Gene is the mechanism of switching off or switching on. Of the genes depending on requirement. So because their expression is not required always. So where? Depending on the requirement of the body or cells, whatever, and how they develop. So therefore, we call these genes as luxury genes. Regulated genes. So these regulation, so this gene regulations are of two types. Negative gene regulation, positive gene regulation. So gene regulation, how is it regulated? So it's of two types, like how enzymes, hormones are negative feedback and positive feedback, that way. So this is negative regulation. So negative gene regulation is repressible regulation. Repressible regulation. And this is a negative regulation. So what is positive regulation? Inducible regulation. Is it positive regulation? So here gene remains non-expressed. So what happens in negative? Okay. What happens in repressible? So this is due to the product of regulatory gene. What happens in inducible? So gene remains non-expressed unless and until they are induced to do it. Okay, so particular product is reproduced and that is a biochemical. So how it happens, let's take a example. So in prokaryotes, so here in examples in eukaryotes, what happens? Gene regulation is at different levels actually in eukaryotes. What are those levels? Transcriptional level, formation of primary transcript. There are so many processing levels. Okay, different splicing, different way of splicing, activation, transportation of messenger RNA from nucleus, all these occurs. So whereas in prokaryotes, so gene regulation happens at the site of transcription and translation. Okay. So there will be, for this regulation, there will be a transcriptional unit. So this transcriptional unit is a RNA polymerase, as you know, at the given promoter gene. So which is regulated by interaction with the different accessory proteins or proteins which are formed. So these regulatory proteins so can both be positive or repressor, inducible or repressible. So functioning of this operator depends on the protein product. So therefore, gene expression is regulated by metabolic, physiological, and environmental condition, and overall these complete set of genes called as operon. So two French scientists. So now let's understand this operon. What is that? So that is operon concept. Many people think that, or students think that operon is a name. No, operon is a set of genes there. Operon concept. Operating concept. See, two scientists, very famous scientists. One is a biochemist, another is a geneticist, Jacob and Monod. So here this name is very famously called. So that is Jacob and Monod. Okay, so that is in 1961, not very old also. So they worked on Escherichia coli, E. coli bacteria. Okay, and they proposed a gene regulation model, and this model is called as gene regulation model is called operon concept. What is this gene regulation model they proposed? See, operon is a coordinated group of genes. So it has structural gene, operator gene, promoter gene, regulator gene. So they all function together. So let's understand what are these structural gene, operator gene, all these. Now let's understand that. So here in the CBSE question, what do you mean by gene expression, regulation of gene expression? They have given 3-4 times. Okay, in 10 years paper, I'm telling you. So I'm just giving you the CBSE paper note also. Okay, so repressible gene regulation is asked always. Okay. So these are the different structures of genes. Okay. Yeah. So let's take up different structural gene. So let me write this. The components of this operon model are structural gene. What this structural gene does? Transcribes messenger RNA for polypeptide synthesis. So operator gene. So this receives the product of regulator gene. Okay, so operator gene is the gene which receives the product of regulator gene. And what is the other function of this operator gene? Anybody? Is it done in your school? Is this operon concept done in your school? Lac operon, is it done?
Yes, ma'am.
So can you tell me what is the function of this operator gene?
And the repressor protein binds to the operator gene site.
Correct. So then what it does? So the complete functioning of the operon, right? It allows the functioning of the operon. Can we say like that? So basically it receives the product of regulator gene. So what is a promoter gene then? These are the 4 genes: structural gene, operator genes, promoter gene, and regulator gene. So what is promoter? Promotes. Promoter, we did it in the transcription also. Now, what is that promoter? Promoter is a site binding site for RNA polymerase. So it forms a binding site. For RNA polymerase. Then regulator gene. Regulator gene. What it does? Synthesis of the regulator protein. So what did the regulator protein does? This is, everything is based on this. Am I right? So everything is. So regulator gene helps in the synthesis of protein. So this regulator protein. So regulator gene, synthesis of regulator. So what it does, regulator gene. Regulator protein is synthesized by this, and this regulator protein itself act as inducer or repressor and controls the complete operator gene. So it helps in formation of regulator protein. Okay. So now here with respect to these 4, so now what is that repressor or inducer? So now let's understand with respect to that different expressions of this model. So first one, let's understand lac operon. So lac operon is, lac stands for what here? Lac stands for what?
Lactose.
Lactose. So is it inducible or repressible? Lac refers to lactose. Is it inducible or repressible?
Repressible.
Why? Okay. Now I'll explain, then I will fill up this line. You will tell me, is it inducing or repressing? Okay, let's understand this. So lac refers to lactose. So in E. coli, in E. coli bacteria, so lactose breakdown requires 3 enzymes. So all these 3 enzymes are synthesized in a coordinated manner by the functional unit of DNA. Okay, so which is called as lacoperon. So what is lacoperon? So breakdown of lactose requires 3 enzymes, and all these enzymes are in a coordinated manner. It is synthesized. So addition of lactose itself stimulates the production of required enzyme. So is it inducible or repressible now? Tell me. Addition of the lactose itself help in the formation. So here there are 3 structural genes. Which are those 3 structural genes? Which are those 3 structural genes? Arya, I want Arya to answer all these. Which are the 3 structural genes, Arya, here? Arya? Okay, Momita, Arya or Momita, one of you answer dear.
Could you repeat the question?
Okay, so what is that we are talking about? So lactose means lac means lactose. So we are talking in prokaryotes. So it's worked on these people worked on E. coli. So in E. coli, breakdown of this lactose requires 3 enzymes. Which are those 3 enzymes? I'm asking. So these are okay. These structural genes. What are structural genes? So structural genes are those genes which transcribe messenger RNA or a particular polypeptide synthesis. Polypeptide means these enzymes. Polypeptide means those proteins which are the enzymes. So which are those genes?
LAC Z, LAC Y, and LAC A.
Exactly. So LAC Z, let me write this. LAC Y, let me write this, and LAC A. Let me write this. Just a minute. 1 minute. 1 minute. Can you see this? Yeah. So what is this LAC Z stands for? What this LAC Z stand for? Come on, tell me. LAC Z. Code for what? Beta galactosidase. What it does? Beta galactoside, which is primary enzyme responsible for the hydrolysis of lactose. So lactose is a unit of galactose and glucose, you know that. So it's a primary enzyme responsible for hydrolysis of lactose. What is this LAC Y stands for? So it codes for permease enzyme. Permease enzyme. So it increases the permeability of the cell, okay, to beta galactoside. So increases the permeability. So in the cell, beta galactoside should go. So permeability is increased by this particular enzyme. What is this LAC A? LAC is a gene which codes for another polypeptide that is an enzyme, transacetylase. Transacetylate. So this transfer acetyl group to beta galactosidase. Okay. So transfer acetyl group to beta galactoside. So all these 3 are the structural genes that are present. So from acetyl-CoA to beta galactosidase. Okay, so it is transferred. So these are the 3 structural genes. So such a structural gene, each of these structural gene of an operon are called as cistron. So this is another important point for NEET. Note it down. Each structural gene of these operon is called cistron. Is that clear? Structural gene. So now, what is next? So we have understood here. So what are the products, components here? Operator gene. So which has to receive the product of the regulator gene. So let's understand this operator gene here. Which is that operator gene? So operator gene is a gene which interact with a protein molecule. So where is it present? So this operator gene is present here. O for operator. So this is the operator gene place on this. Okay. So it lies adjacent to the structural genes. So what it does, it controls the synthesis of messenger RNA. Operator gene controls lies adjacent to structural genes and controls the synthesis of our messenger RNA. Messenger RNA over the structural genes. Is that clear? So that is operator. Next is promoter gene. So what is a promoter gene? What is that we told about promoter? It's a binding site for messenger RNA formation, that is RNA polymerase. So where is this promoter gene? So it's a binding site. So it has to be next to this. So promoter gene, the place for promoter gene is this one. It is this stage. Next is regulator gene. So regulator gene is a gene which codes for a particular protein. So this protein is known as repressor protein. Okay. Oh my God, this people. Can you see the screen, guys? Can you see the screen?
Yes, ma'am.
Yes, ma'am.
Okay. So now we were talking about, operator gene over promoter gene. So what is that promoter gene? Promoter gene is a gene where it's a binding site. So already we have told that it is a binding site of RNA polymerase. So what happens when the operator gene is turned on? Enzyme RNA polymerase moves over it and reaches the structural gene to perform this transcription. That's what will happen. So next is the regulator gene. So where are these regulator genes? So regulator gene, so we call it I here. What that? Oh God, this people. Am I getting disconnected? Is it, students?
Yes, ma'am.
I am not understanding. The moment somebody starting the Zoom, I mean, other class, 8 o'clock, 8 to 9, there are so many classes actually, 1 hour classes for 10th standard. So the moment they are starting, I am getting disconnected here. Each one of them are starting and I am getting disconnected. I need to figure out why this is happening. Okay, I'm very sorry for that inconvenience. I've joined again. Please, yeah, the flow is interrupting. Okay, so the regulator gene is the gene. This codes for a particular protein called repressor protein. So this repressor protein, so what is that protein? What happens? It binds to the operator gene so that the transcription of all these structural genes stops. So repressor has two binding sites. Which are those binding sites? Operator gene and co-repressor. So what will happen here? What is happening here? So, come on. So it's the binding site for coding for repressor protein. So this repressor protein is synthesized all the time from this gene I. So that's why, because it is produced all the time, it's a constitutive gene which is functional all the time. So therefore, repressor protein, and this is a constitutive gene, housekeeping gene. So where is it present here? It's present on this site, gene I. So now, this is how it continues. So now what is happening now? Now, Momita, can you explain this? Let's see.
In the presence of lactose, the conformational changes take place in the repressor protein so that it cannot attach to the operator site, and hence RNA polymerase can slide over and transcription from the structural gene takes place. But in the absence of lactose, the repressor protein binds to the operator site and hence blocks the RNA polymerase from sliding over the structural gene, and hence transcription does not take place.
Exactly. So let me explain this. So let me copy and paste one more here. Okay. Okay. So if lactose is provided into the bacteria culture, that is a growing medium, bacteria medium of this. So then what happens? The lactose enters into the cells. So how? Through the action of permease. So then what happens? So very low level of expression of this lactose operation had to be present in the cell all the time. So what happens? Otherwise, lactose cannot enter into the cell. So in the absence of this inducer, such as lactose, allolactose, all these act as inducers. So then what happens? So the repressor is inactivated by interaction with this inducer. So then RNA polymerase access. So this particular thing will this will trigger the RNA polymerase to the promoter and transcription proceeds. So here we can say repressor binds to the activator. So repressor binds to operator. So what happens? Repressor binds to operator. So this produce, I gene produce, repressor, let's say, messenger RNA, and this messenger RNA produce a polypeptide, that is a repressor. So this is repressor. So now this repressor, it binds to operator. When it binds to operator, what happens? So it prevents the RNA polymerase from transcribing the operon. Very simple concept. Prevents transcription. Let me write that way. That is easy. Prevents transcription. Operon transcription. So that means in the absence of this inducer. So this acts as an inducer. So now repressor messenger RNA is formed. So what happens in the next scenario? Can you tell me, any one of you? Varshini, can you tell me here what happens here in this case? Repressor is formed. So now this repressor is inactive. So this inducer will bind to this repressor. So what happens now? This is free. If then this is free, this will produce all the three enzymes: galactosidase, permease, transacetylase. So this leads to translation. So this translation, so that is beta galactose. So enzyme 1, enzyme 2, enzyme 3. So this leads to lactose to produce galactose. That means breakdown. So glucose and galactose. So this happens. Is it clear? So now tell me what is happening with respect to this lactose here. So lactose is acting as a repressor, as an inducer.
Inducer.
Inducer. So is this positive or negative?
Positive.
Are you understanding? So this is an important concept here. Is that clear? So is the lac operon a positive or a negative? That is what they will ask you here. So it is an inducible operative system, and an inducible operating system is always a positive or negative?
Positive.
So why do you say it is positive? So when sugar lactose is added to the E. coli, so they enter into the bacterial cell for the action of the enzyme permease. So the small amount of this enzyme permeates in the cell even when it is not working. So these few lactose are converted into the active form. So they act as inducer and bind to the repressor. So the inducer repressor complex fails to operate this. So then it is turned on. So when it fails to operate, it is turned on. So now 3 genes are produced. So actual inducer is allolactose. Allolactose is in, it's an isomer of lactose. So you can write that important point. An isomer of lactose called as allolactose is the actual inducer here. Real actual inducer. So because it is easy to say lactose, we are saying actual inducer is that one. So this is all about that. So just depiction of this picture would help you. Okay. So depiction of this picture would help you. What happened to this half? Okay. Is this clear? So now let's go to the repressor. Repressible, that is a negative. So what happens here in repressible system? Tryptophan operon. So this is also an E. coli and it is a repressible. So now tell me what happens here. Anybody? I want Arya to tell me this. This is an easy one. Again, Arya, over to Arya. Tryptophan operon. Come on. So here also 3 necessary enzymes. Which are they, Arya? Which are those? Addition of repressor, addition of lactose induced the production of enzymes. Here addition of tryptophan stops the production of enzymes. So there it is induced. So you can write addition of tryptophan. Tryptophan stops the production of production of enzymes. That's why it is repressible. So this is basically in an anabolic pathways. What happens here? So come on, structural genes again. So let's write the structural genes. Which are the structural genes here? So here the structural genes are, so oh my God, what is happening? Okay. So tryptophan operon has 5 structural genes. These are A, B, C, D, E. So A, let me do it small. So A, B, C, D, and E. So it is A, B, C, D, and E. So that is the 5 steps of tryptophan synthesis, basically. Enzyme A, B, C, D, E. So operator genes. So what are operator genes? These are structural genes. Let me write, these are structural genes. Okay. So you want me write? Figure would help you, right?
Yes, ma'am.
Adjacent to the structural genes are operator genes always, which controls the functioning of these structural genes. So which are those operator genes here? What is operator gene called? Always. Oh. So this operator gene is always kept on. Why? Because the aporepressor produced by the regulator genes is unable to completely block this operator gene. So this is one point you need to remember. So here, operator gene is always switched on. Why? Because the aporepressor that's produced by the regulator gene is unable to completely block operator gene. So when this operator gene is switched off, so when does it switch off? It will switch off whenever co-repressor is available along with aporepressor. Therefore, it is switched off whenever co-repressor is available along with aporepressor. This is very important sentence. So along with aporepressor, co-repressor is also required. So now next is the promoter gene. So what is that promoter gene? So promoter gene is a binding site for RNA polymerase as we know it. So this is here. That's a promoter gene, which is for promoter. Is that clear? This much? Yeah? Okay. So what is an aporepressor? So I'm talking about aporepressor, co-repressor. What is that aporepressor? Anybody? What is an aporepressor? What is a co-repressor? Tell me. Anybody? Momita. So aporepressor is a okay. What is co-repressor, Momita?
Is it similar to apo enzyme and coenzyme?
Exactly. So what is the apo? End product of reactions. So whatever is produced, catalyzed by enzymes produced by the structural genes. So aporepressor is the regulatory protein synthesized by the regulator gene. So when this co-repressor substrate is available in the site, so the aporepressor combines with the co-repressor to form an aporepressor. 1 second, I'll show you the beautiful depiction of this I have with me. One particular picture I would show in that, so you will understand much better in that. Just a minute. So where is it? Yeah. Give me a minute, I will show you in that and explain in that, and I can give you that picture only to understand. So in the PDF.
Ma'am, are you speaking?
Ma'am, you're on mute.
Okay, I'm sorry. So here, repressor gene, this one, tryptophan, so encodes for these. These are the structural genes. So these are the structural genes, A, B, C, D, E. Okay, so these are the enzymes. So that is required for the tryptophan synthesis. So what is happening here? So here you can see, so these structural genes, so they lie in constitution synthesize enzyme. So that is in the 5 different steps, 5 steps. So operator gene, which is lying adjacent here to that, controls the function of these structural gene. So it is kept always switched on. Why? Because the aporepressor produced by the regulator gene does not bind to it, and the operator gene is switched off when the co-repressor is available along with the aporepressor. So that is what we can make out here. So now, what is the how it operates, this tryptophan? So when tryptophan is high in the cell, then it binds with the repressor protein and change its conformation so that it becomes active and bind to the operator near the promoter. So binding of repressor protein to operator overlaps the promoter. So RNA polymerase cannot bind to the promoter. Hence, transcription is stopped. So since repressor tryptophan is already high in the cell, no transcription of structural gene is required, because here it is not the breakdown, but it is the synthesis. So therefore, we call it as a negative regulation. Did you understand? This is what will happen here. So here the aporepressor is a proteinaceous substance synthesized by the regulator gene. And it's a component of repressor for blocking the working of operator gene. Blocking the working of operator gene. Are you understanding? So what is co-repressor? It's a non-protein. Aporepressor is a protein. So please write this particular point. Aporepressor is a protein. So when tryptophan is high in the cell, what is happening? So when it is high in the cell, so please understand, when tryptophan is high in the cell, it binds with the repressor protein and change its conformation. So all these spellings are on. Conformational changes means structural changes, conformational changes. It changes the conformation. So when it changes the conformation, it becomes active and bind to the operator near the promoter. You can see here. So when it binds to the promoter, so then what happens? So it overlaps the promoter. So no RNA polymerase cannot bind to the promoter. Hence, transcription does not happen. So when it is high, so then it is stopped. So here it is a synthesis of tryptophan. There it is breakdown of lacto. Please understand. Okay. So inactive repressor, when tryptophan is absent here. So then operon is open. So when operon is on, messenger RNA is produced and tryptophan. So tryptophan is the non-protein substance. Is that clear? So this exerts a negative control. When it is high, it stops. When it is not there, it is produced. So when it is high, inhibition takes place. So that's what we have understood with respect to hormones also, which is a negative control, negative feedback. Are you understanding? So co-repressor is a non-proteinaceous tryptophan. So how is this inducible operon and repressible operon different? So in inducible lactose operon, so this is not required, attenuation is different. Yeah. So in inducible operon, what is happening? So it is an operon where always it is switched off. Repressible operon, always it is switched on. Are you understanding? So let me write that here. So very important, when you know the differences only, you understand it better. So let's do the differences between these two and wind up the class. So come on, one of you give me the difference quickly. Amrita, can you give me please? Try it out. Amrita? Amrita Varshini. Amrita? Do you give it or not? Okay, who wants to give it? Arya, would you try? Inducible and repressible. Inducible is lac operon. Repressible is tryptophan operon. Okay, so this operon is remains switched off. Remains switched off. This remains switched on.
Ma'am, the inducer binds to the and the other one co-repressor binds to the repressor.
Yeah, so inducer has a prominent role here. So when it is switched on, presence of inducer, it is switched on. Here, inducer has no role. So now, okay, next, what is the difference? Induction of this. Induction starts translation, transcription and translation. Starts translation, transcription and translation. Here, repressor, repression. This starts, here it stops. Stops transcription and translation. So here, transcription and translation. So here, it is operative or functional in catabolic pathway because it is a breakdown of lactose. Catabolic pathway. So here, functional in anabolic pathway, building a process. Here it is synthesis of tryptophan, there it is breakdown of lactose. So here, induction is due to new metabolite. So newly when it is formed. Here, repression is due to excess when it is high. It is repressed, stopped. So here, induction is due to new metabolite, which is formed. Here, repression is due to excess availability of whatever is remaining, that is existing metabolite, we can say, excess. Existing metabolite. Okay, so I'll give you one beautiful picture. I'm not getting it. I had downloaded that. So from one of the IG book. So it is a IG curriculum book. So it's beautifully given the picture. So just by looking at the picture with the diagram, simple diagram they have given. So to make you understand, it's not to follow. So just for you to understand the tryptophan operon, it is given. So I would attach that here. So then you will get to know. So I'll search for it and attach that. So then it becomes easy for you, I think. So here what happens in the promoter gene. So here in the regulator gene, yeah, only aporepressor inactive. Only aporepressor, it becomes inactive. So when aporepressor along with the co-repressor, which is a co-repressor itself is the tryptophan. So when the aporepressor and the corepressor is present, so what happens? Come on, what happens? Guys, what happens when aporepressor and corepressor is present?
The operator is switched off.
Yes. So when both are present, okay. When both are present, so aporepressor and corepressor. Corepressor, which is tryptophan. So when both are present, so then operon off. So here operon on, so it is off. So here it is on. Okay, when tryptophan is present, it's off. When lactose is present, it's on. So that's why that induces this represses. Is that clear? So we are left with, in this chapter, we are left with Human Genome Project, which is a simple, very much simple one. And DNA fingerprinting, Southern blotting. Okay, Southern blotting, we can do it in biotechnology. So DNA fingerprinting is the one. Human Genome Project is the other one. So which requires nearly, I think 40 minutes or nearly an hour. So soon after that, we will take lots of questions, important hot questions I have figured out, and I have made around 50 important questions, which are tough. So let's crack all those, solve all those questions, and solve in the sense not to just give the answer. You have to give the explanation for each of the questions. I am not giving. So 1 hour I'll teach you, and another 2.5 hours you will solve these questions along with some of the hot CBSE questions also. So we will do in the next class and wind up this lesson properly. Okay, thank you all of you. Good night.
Ma'am, could you please share notes from last class?
Okay, I'll share. So last class notes you require?
Yes, ma'am.
Okay, okay, I'll give you. Yeah.
Thank you, ma'am. Have a nice day.
Thank you. Oh my God, Arya is active only at the end always. Okay, thank you.
Thank you, ma'am. Good night.
Thank you. Good night. Vihan, good night. Vihan, please get your mic checked and get it repaired. Okay. Thank you, all of you.